Hi everyone, Russ just let me know that he might be a little bit late, but he'll join us very soon. Hello, everyone. Hi, Russ.
Hey everybody. Sorry I'm late. Thank you so much for having me back. And of course, it should have been obvious to me that it was the same Zoom link. I don't know why I struggled with this. I apologize. Thank you. Okay, cool. Vladimir, should I just get started? Is that okay? Since I've already run
Yes,
a
I think everyone is ready.
cool. Okay, I think you need to stop sharing for me to be able to do it. Awesome. Okay, so we'll go to our Chrome and we'll go back to here. All right, so hopefully you're all seeing some slides. I will, so today I'm going to talk a little bit about kind of the work we've actually done, right? So the last time was mostly about setting up how genomics has been sort of instrumental in studying SARS-CoV-2 and particularly the phylogenetics of SARS-CoV-2 for tracing the spread, locally and globally. But now we're going to talk a little bit about kind of some algorithms to do this at speed and scale and kind of how we got this work to develop. So that's today. And then, of course, next time I'll talk a little bit about recombination and selection in SARS-CoV-2, both approaches to identify and measure it, and then also some things I want to do or that we're working on with you guys to do and we're really excited about. So today I'll tell you a little bit about, this slide has been cut off, but anyway, review a little bit where it came from, some important points, talk about our work studying data and data quality issues in SARS-CoV-2, and then some other issues as they come along. So this is exactly a slide from last time. Just a reminder, zoonosis is incredibly common as far as we can tell. It's probably much more common than we guess, in large part because the vast majority of zoonotic events don't take off, right? They don't have pandemic potential, which is to say they have to be pretty transmissible within humans in the first place, but not so damaging to the host that the individuals die and don't transmit the virus, or that they're so damaging that we lock down areas and just don't let it out. So like Ebola is going to have a lot of trouble with pandemic potential because it's just so damaging to the host that if you had an outbreak in some city, you would simply shut down that city and you would never let it out. But SARS-CoV-2 is this tricky thing that's like a little bit bad, and it gets into enough people to kill a lot of people, which is really unfortunate. So the other main thing that's going on is this is maybe the most disaggregated, that is widespread sequencing effort ever. So I don't think I'm exaggerating to say that, right? What's happening here is that your data is coming from literally everywhere across the world. This is a reasonably recent screen grab from NextStrain, where you can see that, basically everybody is sequencing at some rate. Even I think this is probably Reunion Island over here in the Indian Ocean is sequencing at a fairly high rate. Kind of everybody is. And that means a couple things, but for the purposes of this next section, it basically means that a ton of non-expert people, so people that have maybe never even done wet lab work before, are now making coronavirus genomes and sequencing them on the fly and uploading those data, which means that the data quality varies dramatically depending on who's making it and where it's coming from. And so it's a really important thing to be watching as far as I can tell. And so a really important part of this that we've discovered, sort of indirectly discovered, but I think is really important to think about, is the Arctic primer scheme, right? So almost everyone uses this scheme. There's a few other approaches, but almost everyone's doing this. And this is a screen grab from the UCSC genome browser, where what we're showing you in this top track here is the V3 Arctic primers. Actually, V4 has just been released, and I should also have shown you that, but it's similar, at least in concept, so I think we can work with it. The way this basically works is you have a forward primer here and a reverse primer here. And you can see they also span two other primers. The idea being that you get one amplicon here, and then this one spans to this one. This forward here spans to this reverse here, and another amplicon here. So they overlap, the two amplicons overlap just a little bit in the center. When I do my amplification reaction, they overlap just a little bit in the center. And so the idea is you can tile across the entire genome using an approach like this, right? So it's these two primers also make an amplicon, which overlaps that one. Literally every amplicon just sort of spans another one just a little bit on the edges, out all the way across the entire genome. That's great, except that these primer sequences are not a part of the genome. But the way PCR works, you will certainly be adding the primer to the molecule you're creating, because you prime off of this and it's incorporated in the resulting DNA strand. And so when I prime the other direction in reverse, of course, I copy anything I added there. And so your primers can both mask variations. So if I don't trim them out of my sequences, they can mask important variants in the genome, right? So if I had a mutation here, this primer would basically overwrite it. And that's true in a number of different sites. And so if you have a specific lab or group that's making these kind of systematic errors where they don't mask their primer sites, or potentially the adapter sequences that they anneal to those, right? So if you're doing Oxford Nanopore, you also add an adapter sequence out here to go this way. For Illumina, you would have a slightly different adapter sequence, but basically there is an additional sequence on the end of the primer as well. So if you don't mask those and you don't remove them from your sequence data, then there's a good chance that you've got a real problem because you start to introduce a bunch of mutations that sometimes will align to my genome. And so what you can get is something like this. And so this is what we're calling systematic errors, where basically if your true phylogeny looks something like this, where there's these two mutations, they're present in all of the, in these 4 samples, these 3 samples here, your tree would look like this. But in your inferred phylogeny, you have these recurrent errors, right? In other words, there are mutations that a given lab produces over and over and over again that draw sequences together because they appear to be real mutations, or at least you might think they're real mutations, right? So Gray drew these two together and articulated a part of the tree that otherwise is not articulated in the true phylogeny. And the same is true down here where we got Black and Gray. It even draws in a sample that is linked by a real mutation because there's 2 down here that draw it down to this batch here. So these can really distort your phylogeny. They're tremendously important. And I think it's something that we probably haven't seen in quite the scale that we're seeing it now before, because the vast majority of genome sequencing efforts before the pandemic were not done by 50 different labs or thousands of different labs across the world. They were done by 1 or 2 or 3 labs. And so their batch effects were really obvious. This is a type of batch effect where one lab group or a set of lab groups does the same thing over and over again. So how to find these? This is one of the problems we started with. So when we started hosting data on the genome browser for people, what we discovered pretty quickly is that there were lots of cases where certain mutations appear to occur a ton of times, right? Like they just were all over the tree. It'd blink on and off in different samples. It's very, very hard to track those things and understand what they're doing because they are, either a highly mutable site or something else. And so when we started looking into these, we started with what's called the Parsimony Score, which is essentially the number of times that we think a mutation has occurred across the full phylogeny. If we take the phylogeny and we record the number of mutations, that's your Parsimony Score at a given site. And we record them for each individual SNP, right? So this would be just A at position 3778 to G. And that's kind of how this recording works. And so what we do is we say, okay, or what we did to do this is we said, okay, let's try to break this down a little bit. We have a set of mutations that appear to be highly recurrent. In fact, they have this really nice line it makes. And I think I have some theories as to why this is true. It actually is certainly the case in many other genomes as well. But they have this really nice line they make. And so it's pretty easy to pick out at least the most offensive set of such mutations from these kinds of analysis, where I'm showing you the alternate allele count here. So in other words, how many times we observe that SNP. And this is log scaled so that you can actually see it, basically. And then the Parsimony Score here, how many times a mutation is inferred to have occurred versus the number of samples that have that mutation, basically. Where you would expect that if a mutation occurred a ton of times, at some rate, it must be that they would occur in the exact same, in samples that are very closely related to each other and be grouped, right? So we wouldn't expect this to be a linear relationship. We expect it to be, I think this is log 2, if I remember right, but there's a few different ways you can do this and get a nice clean relationship. Okay, so what I'm doing now is I'm showing you kind of the breakdown. Like if we look at each of these mutations and we ask what's special about them. So in red, I'm showing you the subset that were very strongly associated with a particular lab or a sequencing group. The basic point being that these specific mutations occurred probably because of primer trimming errors in most cases. In fact, this overlaps a primer site, and I'll show you that in a second. So does this one, a few others. There's other examples here that are a little bit harder to understand. And at the end of it, we discovered that what's really going on with these is probably a true mutational process, right? These are all CDU and GDU mutations. Not only that, but they're in a poly-U track. So probably what's happening here is that there's hypermutable parts of the SARS-CoV-2 genome that are just toggling back and forth between G and U or C and U at an incredibly high rate. And we have some theories on why that's true. I think this is related to the thing I talked about last time, which is the host RNA editing processes that seem to be going along in these genomes and sort of interacting with this. And then we have a few, this is the other highly recurrent, which is also a CDU mutation. We see a lot of CDU mutations, as you might expect. Down here is the other way you can kind of think about this, where I've structured this such that it's sort of the number of lineages where I'm ordering it such that each mutation has a certain number of descendants. I don't know why stuff's happening to my slides. Each mutation has a certain number of descendants, and you can see that they quickly level off to very, very small clade sizes of 1, and of course the line stops when it goes to 0. So almost none of these are very big clades, with the exception of these two, C14408U and C241U, which are actually two of the earliest occurring mutations on the phylogeny, and those are probably genuine mutations that got to very high frequencies. So I would say this generally makes sense. Okay, so the other thing that goes on here is that, as I alluded to, probably the biggest source of these errors has been primer trimming and proximity to primers. So there's cases where some are literally overlapping primers, so that's here, here, and out here. There's also cases like this one where it isn't obvious why exactly this happened, but it certainly has a lab-associated effect, and so we recommend it get masked as well. And we maintain a list of masking recommendations for the community because these things have become more abundant over time, and in fact, as more variation has evolved, what's actually happening now is that particular amplicons drop out. So that is to say that the amplicon that's supposed to have occurred between 12 left and 12 right here, sometimes one of these primers has a mutation in the primer binding site, it can't amplify. And so you literally get, you don't get any signal from your true genome, but if you have a low rate of contamination, you do get signal from potentially your positive control or something else, and cause there to be kind of spurious variation in these tracks. So we did some simulations just to see, did we get numbers that make sense, basically. And so what I'm showing you here is what happens when I do, when I just add each of these mutations at these frequencies. And you see this nice strong log to relationship. So this is what we call the extremal, where basically there's a lot of samples that live out on the edge there. And I'm showing you also on here the real mutations that we see in each of these cases. So they're not exactly the same, but they're highly consistent with what we're observing across this thing. And of course, if you do 2 mutations that are highly correlated, you see a much stronger effect because they'll draw each other together, because any tree inference software will think those 2 sites are independent, even though they're not. And the same sort of effect as we observed originally. This is the same basic plot of, you get a couple of really big looking clades, and then they descend into very, very small clades very quickly, as we would expect if there's reasonably frequent systematic errors. Okay, so if I show you an extreme example, oh my God, struggling here. Okay, if I show you an extreme example, this is a case in samples from Belgium, who was a group that didn't trim their Oxford nanopore adapters, basically, where I'm showing you a link between the same samples and a phylogeny inferred, including 3 positions that we think are systematic errors. And in a phylogeny, we have inferred having removed those 3 and just showing you the most informative sites down here. What you can basically see very quickly is that these systematic errors have been a major source of problems, right? Because they're drawing, like, for example, these 2 samples are drawn right into the middle of this tree structure. And the same is true of many other samples as well, right? So there's a lot of cases where they're being forced in, and they're also generating apparent back mutations, in large part because of the same thing that's going on here, where these samples are drawn into the center because they share these systematic errors. So you can really distort your phylogenies, and it can really mess with the way that we interpret this variation. So it's something that's tremendously important to be watching out for. Okay. With that said, another thing I alluded to at the beginning here is that probably a lot of the highly recurrent mutations actually are real. We think there's probably parts of the coronavirus genome that are simply highly mutable, I think is a straightforward way to say this. And you can kind of tell it a few different ways. One is just the CDU versus non-CDU mutations, where basically what you see is that the CDU mutations are much more likely to have occurred 2, 3, 4, 5, 6 times, the Parsimony score here, than our non-CDU mutations. So there is something truly hypermutable about CDU. And the same is true if we take this a little bit differently and we say, okay, but if we think they're real, then these should be subject to selection. And we typically consider non-synonymous sites as more likely to experience selection than synonymous sites. That is, sites that alter the protein sequence are non-synonymous. Those that don't affect the amino acid are synonymous, or at least non-amino acid altering in some cases. And so if you do that, you'll see there is this weak effect as well, where the subset of sites that don't have a strong effect tend to be slightly more recurrent than the subset of sites that do affect the protein coding sequence. It's a much weaker effect, but it's also clearly apparent from the data. Okay, and of course, I think I showed this last time, but just to remind you, so we also did a detailed updated analysis on this, where we looked through all possible positions to find how frequently they could occur. So in other words, the green bars indicate how frequently these positions could occur in both synonymous and non-synonymous positions. And what you can basically see is that there's another type of mutation that's actually extremely recurrent as well, is GDU. So these red bars are much higher than green bars in both of these cases. That means it's occurring at a much higher frequency than we would expect by chance. The reason we didn't see this in the first analysis is basically that GDU mutations are much more likely to alter the amino acid sequence, therefore more likely to experience selection and much less likely to be picked up when we do this kind of analysis. Whereas CDU typically is less likely to alter the amino acid sequence just because of the way the codons are structured. Okay. Even if you had perfect data, which no one does, phylogenetic uncertainty is sort of rampant. What I'm showing you now is an analysis we did of next strain trees across about a month, where what I'm doing is I'm taking the trees, I'm creating a distance matrix between them, and I'm using principal component analysis to sort of take that distance matrix and turn it into something at least somewhat interpretable. And what you can see is that there are certain time periods where you'll live in different parts of tree space. They actually group in kind of interesting ways. The first two coordinates explain a ton of the variation, so it seems like it's sort of a good enough representation, although of course there are people that will argue with me about that. I don't think it's worth going into in greater detail. And so what's basically happening here is as new data are added, it alters the tree structure. Of course, this comparison is done only considering the set of samples that are present in all of these trees. And so it's kind of a bizarre and interesting thing that's going on here. On the other side is just a different way to show this, where it's kind of a tree of trees approach. So we take this distance matrix, we turn that into a tree, and we label them with the dates. And you can see that, yeah, nearby dates, of course, group together as you might reasonably expect. So for example, these are all from March 23rd, 25th, 26th, 24th, et cetera. But then also it does change over time. And so you get different groupings, and it displays a very similar pattern as you see in panel A here. So all of that is to say that even though there are these weird systematic errors, even when you remove them, their effect, there is still a dramatic impact of phylogenetic uncertainty. There just aren't that many mutations in these trees. It is very hard to make nice, consistent phylogenies because we just don't have much information to work with. And so that's the fundamental challenge we're facing here. So as a brief aside, we very quickly realized that we hated all of the ways for making tanglegrams. So a tangle-gram is this thing where I take two trees and I display them side by side like this, and then I draw a line between the shared samples, right? So the same sample would be connected by a line between the two trees. We discovered very quickly that we hated the way these things were made. And so what we decided to do was write our own algorithm. I'll quickly describe that algorithm because it is kind of cool. It's very simple, but it works remarkably effectively, and it makes nice GIFs like this at the end. The basic idea here is that when you have your starting position, which you'll flip back to in a second here, there you go. Obviously, one tree is made entirely without the other, and they're completely unstructured with respect to each other, other than in as much as they have the same basic structure because of the mutations that are present in the tree. And so what we wanted to do was to figure out how to make this, how to clarify what we're looking at, because it helped us visualize what was different when we messed with the trees. And so what you see here more than anything else is, and of course, sorry, to indicate how to read this, at the end you see the best orientation we came up with, which is essentially as many parallel lines as possible crossing this with as few crosses as possible is the best way to display these data typically. And so we found very similar effects between them. At the end, you see these trees are actually quite similar to each other, even though at the beginning you might say, oh, these are totally different. When you move stuff around, you can see it's the same. Anyway, the way this algorithm works is you start out at the root of the tree and you descend the first level. And then for all the samples descendant from that first node, we ask in the other tree what their positions are. And then we sort based on this. Is that one of you? No. Okay. We sort based on this based on their relative ranking in the other tree, right? So I take this set of samples, I say, what is the ranking of those samples in the other tree? In other words, how are they related to each other in this tree? And then I sort this set that way, and I descend from the root down, and I apply all these moves. And then I do that on the opposite side. What I do is I go up the other side and I say, okay, I'm going to do the same thing. I'll descend from the root and I'll sort based on the other tree. And we've discovered that, and I don't think this can be formally proven, although I'd be interested to know, in about 3 iterations, you've exhausted your possibilities and you're done. And so that's kind of how we came to this. It's a really fun tool. You can check it out on our paper or our GitHub page. I find it to be a really, really useful way to compare things when I make, otherwise very similar trees, but try to figure out what's changing between them, because it's incredibly hard to visualize these things any other way, as far as I can tell. Okay, so that's a brief aside. The effect of systematic errors is actually typically very small. So what I've done here is I've shown you a tree inferred, including the systematic errors on the left here, and then a tree on the right where I've excluded the systematic errors. In other words, I've masked all the positions in the genome that I think are systematically problematic because they are present because they come disproportionately from a single sequencing group and they're highly recurrent. What you can see is that these two trees were extremely similar, which is good, right? It means that essentially you are almost getting the right answer anyway. All that was happening is cases where these two samples, for example, are drawn together, and presumably they're drawn together because they share a systematic error. And the same is here, right? This subset all shares some systematic error, hence they actually go to 3 different places on the tree, right? But they get drawn to this one place. And so the other way to think about this is to say that, yes, systematic errors were important and continue to be important, but what's really going on here is that phylogenetic uncertainty is just rampant, even if we had perfect data. It's just very, very hard to find the optimal trees when we do this kind of analysis. Okay, that was a very long lead up of something that I think is tremendously important, which is how do we actually make our trees and what are we doing here? So the phylogenetic inference typically uses maximum likelihood. I think the vast majority of people doing it are using maximum likelihood approaches. The basic idea behind this, I'm only going to explain it briefly because I'm actually going to go to a much simpler thing, which is what we do. But the basic idea behind this is that you want to find the parameter values that maximize the likelihood of the tree. So these parameters might include things like the tree topology, the branch lengths, and the mutation rates. And so it's actually a pretty easy problem if you have a fixed topology to just optimize the mutation rates and branch lengths. It's a much, much harder problem if you don't know the tree topology and you have to search tree space, because what happens is you have to move stuff around, recompute the likelihoods, and it could take quite a while. So the basic idea is, so this is the Felson sign pruning algorithm explained very, very quickly. It's the way I think everyone computes likelihoods. I'm not sure anyone's done anything else different, although I have some friends that are working on this now. But the basic idea is you start from the tips, right? These are the known states, the data we've collected with some sequence. And what you do is you go up the tree, right? So step 1 is just going up the tree to this first node here, et cetera. And compute the probability of each internal node state, ACGT, in this DNA, right? And you can do this via dynamic programming to be much more efficient than if you did the sort of stupid thing and multiplied all the way through every single time, because it would be a ton of operations. You can do this independently for all sites, as long as you assume the sites are independent, and just take the sum of the log likelihoods across all sites. And that is actually a really, really fast way, well, comparatively fast way to get the likelihood. The problem for SARS-CoV-2, and the reason maximum likelihood has actually been kind of terrible for SARS-CoV-2, is that when you're computing the likelihoods, what you're doing is called matrix exponentiation. And I imagine many of you are sort of math background and probably know this better than I do. But you basically, anytime your transition matrix looks something like this, so like let's imagine this is a full ACGT transition matrix. The problem with this is, because my branches are so short, the probability of going from A to A on a branch is enormous, right? It's essentially all cases with some very rare exceptions. Whereas the probability of transitioning from A to something else is very small. And so I think probably most of you know this, a basic heuristic rule of matrix exponentiation is if you compare the largest and the smallest values and you find they're dramatically different from each other, you're likely to experience numerical instability. And the reason that basically happens is if you divide very, very small numbers by very, very large numbers, you get a lot of error amplifying through this thing. And it's incredibly hard to do a good job computing likelihoods using the traditional Felson sign pruning algorithm. And it's part of why people are starting to develop new approaches that don't require this, because it's kind of a crummy way to do it. the other main issue, these trees are big, right? If you have n samples and you have a fully bifurcating tree, then there's typically going to be n minus 1 internal nodes in my tree. And so that means there's a lot of math operations I've got to do to compute these likelihoods in the first place. And so for SARS-CoV-2, this has just been inoperable for the big trees, at least. You just can't do it, or it doesn't work well using existing approaches. There's a few workarounds people have come to, but they just aren't that great in general. Okay. So one thing, you probably know this. I hope you guys know this, but if not, I'll walk through it quickly. The way we typically quantify phylogenetic uncertainty is usually what you call bootstrapping. So in other words, with bootstrapping, what I do is I resample from my multiple sequence alignment each site at random with replacement to an equal number of sites I have in my original MSA. The idea being that if I had infinite data that was more or less equivalent to the data that I'd sampled, I would get some representation of the possible realizations of this given the finite data that I have. My MSA has only so many positions. So for example, a first bootstrap replicate might sample AGG, this site, it might sample this site, CGG, might sample TAC, but then here you see that it's actually resampled with replacement CGG, this position, and done a bootstrap on that. And you do a series of bootstraps. And at the end of this whole thing, what I basically do is I count up the number of times where a node that was present in my original data set, right? So this one grouped B and C with this ancestral node here. I count up the number of times, in this case, there's 3 bootstrap replicates. So B and C are grouped in 2 of them. So that would be 66% of the time I group these 2. So that's great when you have a lot of data, and in particular, when you have many, many mutations. So for the vast majority of phylogenetics that's been done to this point, the data matrix is very, very long this way. That is to say, we have a lot of sites, but not that many taxa. The problem for SARS-CoV-2 is that your data matrix is actually long the other way, right? There's a ton of taxa, a ton of different samples, but actually very, very few sites. So, and so when you do your resampling, if there's only one mutation that links a bunch of samples to each other, the probability of resampling in a tree may not be that high. Okay, and in fact, that's the exercise I'm going to leave you with while I get more coffee here. Basically is this question, right? So I've just explained to you bootstrapping really quickly. I'll leave this up here so you'll have this representation. But basically the question I want to ask is, what is the expected bootstrap support value for a node with exactly one mutation on a branch? Not necessarily this multiple sequence alignment here. Just imagine it in your mind. Assume that there are no other site patterns that conflict with this. In other words, that mutation can be considered independently of everything else. So what is the expected bootstrap support value? Do the same thing again, but now think of a node with exactly 2 mutations on a branch. And I think to do this, I'll send you off into small groups, breakout rooms, if you will. How do I do that though? Let me see if I can figure this out. So if you could help me, anybody, that would be great. Otherwise, I'll just struggle through this and you can watch me struggle a little bit. So let's go back to the main one. Can I do it here more? So Vladimir, I'm not sure I can send people to breakout rooms. Can you do this?
Well, you are the host now, so
okay.
I think you should be able to do that.
Okay. Typically, I would do that under participants, right? More.
I'm not sure if I enabled.
I don't think I have the option where it typically lives on my computer. So yeah, there may be a way to do this. Okay, I don't want to keep you guys sitting around while I mess around with the computer. So what I'm going to do is say, do this individually, and we can all come back at the end and talk about it. Does that sound okay? Here we go. I'm going to leave you with this. Thank you so much. I'll be back in a second. Okay, sorry, there's a question from the chat here. So if the question is, could you repeat what the supporting value is? And that's a Bootstrap support value. And what that basically means is, if I take my sites in my original data matrix, and I resample them with replacement to create what's called a Bootstrap replicate, right? So the first one is this replicate, the second one is this. You can see they're all slightly different collections of the sites, right? And so this last one actually sampled only 2 sites and sampled each of them twice. If I take those and I re-infer the tree, what you can do for every single node on the tree, so every internal node, is compute how often the descendant samples are grouped together, right? And exactly that descendant sample. So in this case, B and C is the node we're computing. And you see B and C in replicate 1, in replicate 2, and not in replicate 3, right? In replicate 3, A and B are grouped, B and C are not. And so what you would say is that 2 out of 3 of these, or 66% of the time, we recovered B and C. So that's our Bootstrap support value for that node. And I'll give you all another minute because I feel like I didn't explain that well enough. So we'll come back in a second.
Russ, I just checked the breakout rooms were disabled for this session, so we can either restart the meeting or just use them next time.
Let's just do it next time. I don't think we need, it's not essential. It's okay. We'll make this work. Thank you, Vladimir.
Yeah, sorry, I just.
No worries. We'll figure it out. Okay, let's come back. So does anyone think they got this? I think it's probably simpler than you may think in some cases. Does anyone think they have this? No one? Should I call it somebody at random? Oh, I cannot pronounce at least a couple of these. Let's see. So, oh no, I got all these. Anybody? No? Okay. Well, I don't want to call someone out. I guess I'll just do it myself here. So, so the question was, what is the expected Bootstrap support value for a node with exactly 1 mutation on a branch? And assume that no other site patterns conflict with this, right? So, and the same question again, except for 2 mutations on a branch. What you basically want to think about is what is the probability that we'll draw the site in at least 1 Bootstrap replicate? And so that would be 1 minus Poisson 0 if with an expected rate of 1, about 63% of the time. For 2 SNPs, you'd get the same thing about 87% of the time. And the important point of this exercise is to show that when your branches really only have 1 or 2 mutations on them at the most, which is true of the vast majority of branches in SARS-CoV-2 phylogenies, typical bootstraps are not going to be very useful because we typically throw out anything with less than, say, 90 or 95% support. And so you're going to have a lot of trouble generating high support values for these branches. So again, this is your problem, right? The scale and the pace of the data generation is extreme. We just so much data coming in all the time. You've got very, very short branches that link them because there's a gajillion samples. So in a typical phylogenetics workflow, I'm going to explain what we're doing that's a little bit different from the typical way people do this. In a typical phylogenetics workflow, what you would do is gather the data and calculate the tree. But the problem is that if I want my tree to be comprehensive, and I think that's really important for certain questions in public health, the problem is that as soon as I have finished calculating my tree, it's already out of date because more data has shown up. And so you see my sequence alignment got a little longer. And so you could do the obvious thing of recalculating the tree, which takes a few more days. Or, but of course, if you do this, the same thing will happen, right? New data has now shown up. My matrix is even a little bit longer. And so if I recalculate the tree again, it takes a little longer. And this is a sort of losing proposition where I can never catch up to today when I would have a full and complete tree to work with. So what we've been developing, and we're not the only people to do this, but we are some of the people that are doing this, is an approach that we call online phylogenomics. And basically the way this works is, on the top, I'm showing you de novo phylogenomics, where we do this, where we infer the tree from scratch every single day, right? So you see that this first day is this green tree, the second day is this blue tree, the third day is this purplish tree. It's doing okay. Over here, we have what we call online phylogenetics. And basically the idea is I inferred this green tree on the first day. On the second day, I've simply added in the new blue sequences to my tree, where it's still the same basic underlying green tree. On the next day, I do the same thing again, where I add stuff to my existing tree in purple here. So the way that we do this, why is this so efficient? Well, or why is this better? Well, it's obviously better in the sense that if I start from scratch every time, I have to infer all the other things I already kind of knew about the tree and the new stuff. But also there's a way that we can do this that's incredibly efficient. And what we did for this is an approach that we called ultra-fast sample placement on existing trees, or USHER. And the basic idea behind USHER is this data object here, which we call a mutation annotated tree. The logic of this structure is you have a normal tree, you've got A, B, C, and D here. And what I've done now is, those are your taxa, your tips, right? What I've done now is I've labeled each of the internal branches on the tree with a number, right? 1, 2, 3, 4, 5, 6. And what you have on the other side here, the rest of my mutation annotated tree file, is the location, or is the mutations that occur on each of those branches. So branch 1 here has mutation 6G on it. Branch 2 has 4U, 3, 7U, etc. Right? So each of these is annotated with the set of mutations that occurred on that branch. The logic behind this is that not only is this an incredibly efficient data format to store, for reasons that I'll talk about in just a second, but the other big advantage is what I've done is I've stored the entire inferred evolutionary history of the virus, right? So I've got the complete tree, I've got all the mutations that I think have occurred. This is a really, really, really useful way to go. So if we do that again, but then when a new sample comes along and I want to add it to my tree, what I can do is just brute force parsimony. So initially we wanted to do what's called a tree search, where you would descend from the root and just see what matched. But the problem is there's so many recurrent forward and backward mutations and other things that go on in these trees that this really doesn't work. And so what we ended up doing was just checking every single branch. So we literally check every single branch in the tree, and we say, we identify the one where this sample requires the fewest additional mutations to be added to my tree. That's the maximum parsimony placement. Parsimony is really nice relative to likelihood, because parsimony is a much, much simpler thing to compute, right? It's just it's a matching set problem of how many matches do I have in a given set. And in this case, you can see that sample E matches mutation 10U on this branch here. So it requires only one additional mutation to add sample E to my tree, which I do here. And then I can actually store this, right? So this in memory is still a mutation annotated tree, and I can just write this mutation annotated tree object to disk very quickly. The idea being that now I can do what's called online phylogenetics, right? So I add sample E, I might wait 10 minutes, then a new sample shows up. Well, I'm able to add that almost as quickly because I can just load this new mutation annotated tree file, do the same search across all possible branches in the tree, and add that sample to my tree as well. Of course, as the name would imply, USHER is ultra fast. So the time it takes to place 1,000 sequences on the tree is shown here, where we first actually started this project. I thought, well, this is an old problem. We'll just use some of the existing, some of the existing phylogenetic inference software that's out there for placing samples onto trees. And we had PAGAN2, IQ-Tree2, TreeBest, RaxML, and a few other things. And what we basically found is each of them took more than a day to add 1,000 sequences to about a 40,000 sample tree. And so it just wasn't going to work. And Usher, on the other hand, when we wrote that, took about 43 seconds. So it's many orders of magnitude faster than anything we were aware of. And this has really been sort of the reason Usher is so efficient, is that it has a really efficient way of representing the data and an efficient way of adding new stuff to the tree as well. The big difference is that these other programs, for example, IQ-Tree2, what they're doing is they can do tree addition in the sense that they store a tree, but what they'll do is they'll load a tree and then they'll load a multiple sequence alignment, and then they'll annotate all the nodes in the tree where mutations are inferred to have occurred. Usher stores that information. So we skip that whole first step and it can just go much more quickly. It is also a highly accurate way to do this. So Usher is real good at what it does, in the sense that about 97% of our placements are topologically correct. That is to say, the user sample is adjacent to existing sample 1, or in other words, it has the exact correct placement where the sister lineage is exactly the one we would expect it to be. So it's topologically correct. When it's wrong, it's typically not far off. So Usher sometimes places samples up here at this node, for example, 1 node away, things like that. But it's almost always in the right phylogenetic neighborhood of the tree. So cool. Okay, so what we have then is, so this is returning to this problem of phylogenetic uncertainty. I need to explain how we were doing this. So we don't think that bootstraps are going to work very well. And the reason we don't like bootstraps in this context is that, like I said, they just don't give you a lot of support. You can do other things like what's called an approximate likelihood ratio test. But all of these things are actually not really well suited to this case, where there just aren't many mutations. My data matrix is actually very sparse on each branch. And so what we've been working out is other ways to quantify uncertainty that are a little better suited to public health uses. The other reason we think this is important is that for a public health officer, probably the vast majority of the time, a given sample is, so probably the vast majority of the time, they really care about knowing is, what are the close relatives of my sample? Right? They probably don't care if this internal node is highly supported, because it's irrelevant to them if their sample actually goes down here somewhere. And so what they really want to know is something else. And so there's 2 ways we thought about doing this. The first is what we call the branch parsimony score. And what that basically is for all the branches shown here on the tree, it's how many additional mutations would be required to accommodate this sample placement. With the idea being that you can see that most of the branches down here are pretty good. They require something like 1 to 5 mutations in general. And as you go further out on the tree, you get up into like the 10s and the 15s. And then finally out here is really, really bad placements where obviously it does not go there. The second way we thought about doing this is to record the number of equally parsimonious placements. The basic idea is, there may be many places on the tree where a sample requires the exact same number of additional mutations. And that tells you something pretty quickly about how likely it is to be the right place when I put my sample on the tree. Because if there's 4 places that are exactly equally parsimonious, my chance of getting it right naively is 1 in 4, right? And so we've got a real problem there, and that does happen at some rate. I'll say that at least for SARS-CoV-2, the 85 to 87% of all samples have exactly 1 equally parsimonious placement. And then after that, about 10 to 12% have 2, and then 3 or more is a very small proportion. But it does happen, and that typically happens when you're not making very good data, as far as I can tell. Okay, just to show you kind of where we've come to, the current big tree is shown here. It has scaled really well, actually. So the current thing is about 4.4 million samples, 4.5 now. Each edition still takes about a half a second, and the whole thing, that is the mutations, the tree, is encoded in a file that's 70 megabytes in size. So this is, so again, this is this really wonderful thing that to explain why this is so good, this evolutionary compression. If you can do this for 4.5 million samples at 70 megabytes, I can literally email this tree structure to people whenever I want to. It's just not very hard to share data at scale. And I think that's a really important thing about this, because when you have this extremely disaggregated data generation process that's going on around the whole world, you also need to be able to share those data back. Okay, so I'll show you some comparisons we're working on right now. I think a lot of people maybe rightly ask me, how does the big tree, how is this compared to other methods, right? So many people are familiar with maximum likelihood approaches. And then, of course, we're also doing this other thing of sequential addition and optimization to an existing tree. So these two things are hard. And so what we would do is, sorry, we're showing you here, it's going to take a second to walk through the whole figure, but what I'm showing you is a couple of different things. Normalized parsimony score, where what I've done is I've normalized it to this online mat optimize approach that I'm using, where again, I'm adding sequences to a tree that's online. Each of these that says online is this idea that I add my sequences and then I optimize and I add and I optimize. And I've done this against IQ tree 2, fast tree 2, and our approach, which we call mat optimize. And what you can basically see is that we're doing better. So the normalized parsimony score is just how much worse each of these trees is in total tree length than the way we're doing it. And you can see at the worst up here is 1%. 1% additional mutations is online fast tree 2. De novo fast tree 2 does a little bit better. For whatever reason, de novo on IQ tree 2 is worse than online IQ tree 2. Then you can see our 2 down here, where we either de novo build a tree from scratch and add and then optimize, or just add stuff in line and optimize, is down here. It's doing pretty good. The thing that I think is really important about this is that that's parsimony score. It's literally just counting the mutations. So I would assume I would do better for parsimony score because we wrote a program to optimize the parsimony score. So that seems utterly reasonable to me. But what I think is tremendously interesting about this is that you get almost the exact same pattern. We're not quite done computing this yet, is why it's not finished, but you get almost the exact same pattern if you instead use the normalized log likelihood scores, right? So what we're doing here is we're saying, okay, well, I'm sort of cheating, right? I've done this whole thing with parsimony. That is, the fewest mutations is the rightest tree. What I want to know is if I was to compare to the approach these guys think they're optimizing. So fast tree 2 and IQ tree 2 are both optimizing by likelihood. Would they do better if I do it that way? And the answer is actually no. It turns out that it's extremely strongly correlated with the parsimony score difference, but also that likelihood is just they're not doing as well as we are. And I think the reason for this is not that there's something fundamentally flawed in likelihood, other than it's very hard to compute. The bigger issue that's going on here is that they don't, you just can't search as much tree space as I can, because the comparisons take much longer when you have to optimize several different branch length parameters. Whereas if all I'm doing is counting mutations, I can do that really quickly. So I can search a ton of tree space way faster than these guys can. And I think that's why mat optimize and online mat optimize do so well. The other thing to show you is this. If we just measure the wall clock time, you'll see that the kind of all of these approaches don't do very well. And we cut them off after a day because we said, well, that's not fast enough. It won't work in a pandemic. The only ones that seem to do okay is de novo Usher and then this online IQ tree 2. But again, you know, we're up to about, I think it's 15 minutes or something out here doing a couple 100,000 samples was the total amount of time. Now, you might say this should be represented cumulatively, which I think is reasonable. But the important point is to say that this approach is just way more computationally efficient. So this is memory, same basic effect is going on where, you know, IQ tree 2 is extremely memory hungry. And so it's really hard to run it on basic servers, on big trees at least. Whereas again, we're doing really well for memory usage because this data structure is extremely efficient. So that is to say, we both get more optimal trees. The difference here is not that much, but it is something. And then also we're much faster at doing it. So it's a much more efficient way to do this. Okay. That, of course, is just the parsimony score and likelihood of real data. You might want to know about something else, which is what is the parsimony score or likelihood, or sorry, if we do the same thing, we optimize these ways, but we now use simulated data. And when I use simulated data, the important thing is I actually know what the correct tree is because I fixed it when I simulated my data. And so in this case, I think we used the tree we inferred originally from all the data, but I've also done this using VGSim, which is Vladimir's simulator that I've really, or your guys' simulator that I've really enjoyed. And you get kind of the same answer either way. And so what I'm basically showing you is that, yeah, de novo fast tree 2 is, you know, substantially worse no matter how you look at it. But everything does kind of badly, except for online IQ tree 2, where I add all my samples with Usher and optimize with IQ tree 2. De novo Usher and mat optimize here, but then on the bottom, the best for the vast majority of iterations is this online plus mat optimize, as we might expect. So we think that online, the point of all of this is to say that we think that doing this online phylogenomics using parsimony is, not only is it the only thing that's going to work, because, as I said, you're just going to run out of computational resources, right? This is actually, the largest tree here is only a few 100,000 samples. We're currently up to 4.5 million. I don't need to be spending all my money on Google Compute to try to run these things. And so we're kind of stuck down here at the bottom with using this much more efficient approach. Okay. So this has been, I think, a tremendously important way to do these kinds of things and something fun to think about. Okay. And this analysis, I should say, this analysis is ongoing. That's why none of these lines are done. So we're still running these things. They just take a while to run. But I think that the trend is pretty darn clear and it's unlikely to change a whole lot. And so it's kind of a fun new thing that we're working on and thinking about a lot, because I think it's going to be tremendously important, not just for SARS-CoV-2, but for the future of the field, where pathogen genome sequences are going to be extremely abundant and we need some useful ways to make sense of them. All right, so I titled this slide probably the first question I would get from people, which is, so you've got a tree, now what? So what's going on here is the last mile is what we like to call this in the field. It's the hardest part of this. And what that basically means is, how do I go from taking genomic epidemiology and getting it into the hands of public health officers such that they can take meaningful action based on this information? I think that has been maybe the hardest, most crucial part of the pandemic, and it's something we've made the least progress on, right? There's a lot of people that kind of sit around using these data to tell the retrospective story of how SARS-CoV-2 evolved. I don't think that's anywhere near as important as trying to speed up this process and get it into the hands of public health officers so that they can take action and do something with this information as it comes online. So the questions we're going to work with here is, how can we make this information actionable to public health measures, and can we easily automate some of the analysis? And that's what we've been working on quite a bit recently, and especially this last part. How can I automate this analysis such that I can answer some of your public health questions without you even looking at the data? The biggest questions people ask are, how many introductions were there to a given area? And how big did each introduction get? Right? Did it really take off and become a dominant strain, or was it relatively small? And so when I say introduction, what I mean is the event where a new strain is introduced in the area. That would be represented by this little branch here. And then a cluster would be a set of samples that are descendant from that same introduction event. So all of these things encircled in red here is a cluster associated with this one introduction. And we infer that because they are all sampled in the same geographic area. And so it's pretty easy to work with it. Okay, so some design constraints, right? So that we want to run this daily with millions of samples. So again, you can't be doing this on maximum likelihood or Bayesian approaches because they'll just take way too long with way smaller sample sets. So we want something simpler than that. It also has to be able to handle polytomies, right? So a polytomy is a case where you have multiple samples descendant from the same root. So you don't get a bifurcating tree, you get these much uglier trees where there's a ton of like literally identical genome sequences and stuff like that. And finally, you'd like this to have a simple and intuitive data exploration platform. So you don't want people to sit there and click through thousands of subtrees. You want them to be able to make sense of these data real fast. Okay, so what do we do? Well, we came up with a simple heuristic for this, right? Nice thing about a heuristic is it can be computed once by traversing the tree, post-order traversal of the tree exactly one time, which tends to be a very efficient way to do these kinds of things. So this would be a case, like this is a simple case just to show you how it works, where I have 3 samples that I'm calling in a given region. That is to say that they are sampled in the same geographic area. It could be a city or a state or whatever, or a country, it doesn't really matter. And then everything else would be out, right? So this blue sample here is out. And then this is my focal node. So the rest of the tree is up here. We don't care about that because what I'm doing is a post-order traversal. That is to say, I start at the tips and I move up the tree. Which is a really good way to do this sort of thing, again because it's easy, it's quick, it takes only one traversal, so we're very good at it. Okay, so down here we have one sample, and our heuristic is basically getting a ratio of mutations to number of samples. Mutations being the genetic distance to the most closely related sample to a given node, and the number of samples that descend from it. The idea being that we would like to weight the assumed origins of a node based on how many samples are in or out of a region, but also how closely related they are. Because as you can see, this one's only one mutation descendant from that node. which means it's probably much more closely related in time. But again, there's only one sample, whereas here we have spread into 3 different samples, and you see it's quite a bit more distant. So, but it's more genetically distant. So if we do the same thing, we compute that ratio on the other side, we get a number that's 1.33, right? So the nearest sample is 4 mutations away, and there's 3 descendants. So if we do that, we're going to express this under a squash function, right? So 1 over 1 plus 1.33 divided by 1, so the ratio of the two. And you always get a number somewhere between 0 and 1. It can never be 0 or 1, but I think it can sort of approach the edges very quickly. So 0.43 is what you get here. Again, it's a heuristic, so I can set my cutoff to whatever. We typically use 0.5 as the cutoff. And in this case, you'd say, okay, this node is actually out of my focal region, my in region. But these other nodes obviously would be in because all of the descendants are within this region. So we're going to call that as in, we're going to call that as in, and my introduction would be this branch here. Which seems like a pretty simple, straightforward way to do this. We have tried a few other heuristics, and of course, I'd be very happy if anyone here or anyone in general wanted to suggest others that we could try that would be maybe a little better for this. You know, I'm not claiming this, you know, it's a heuristic. I'm claiming it works. I'm not claiming it's principled. And so hopefully that's okay with people. But the important thing about this is it's so fast that I can do this for many regions in parallel, right? Where I traverse the tree for all samples from several different regions, and I do the exact same sort of thing. And the idea behind that is, well, then I can actually start to identify, in this case, I would say, well, this node is probably actually in whatever region this sample's from. And so not only can I find my introduction branch here, but I can say, okay, this introduction branch is probably an introduction from wherever this sample was sampled from. And of course, you know, my confidence for that would be the reverse of this, 1 divided by 1.33 here. So that should be 0.57, which would not be very confident, but would give me some idea what's going on. Okay, so when we do that, there's of course some issues and tweaks we did on top of this, right? So the first is that you've got nested clusters. So, you know, you have descendants and descendants and descendants. So this is an example of this where all of these might be a single introduction event, right? This node here might have introduced all of these and then the virus spreads. But it must have also spread back out of my region, right? Because you see all these other samples that I'm not circling that may have come back out. And so it's what we do is we spend some time trying to figure out how to merge stuff, particularly when you have genetically identical samples like these. Right? They could all be unique independent introductions, which would be very hard to figure out. And so what we do is we just say, okay, we're going to merge all the identical ones within a given region into one cluster and hope for the best. So I'm not claiming this will give me an exact number of introductions. I don't think anything will, but it's very, very quick. I can do it every day. And the important point behind that is I can then host this entire website and do this at the state level for all of the United States every single day. And they can use what's called cluster tracker to click around and see when there is a new cluster that's been introduced into a given region, how quickly it's growing, where it might have come from, what the variant, the lineage or the next clade calls are, that kind of thing. So that's how I'm going to do this. And it's worked pretty well as far as we can tell. People are enjoying it, and it's been a lot of fun. If you want to try it out, I'll throw, I think I sent this to you guys last time, but I think it's just kind of fun in general. Cluster tracker. I'll throw it in the chat. You can try it out. Right now, it's only for the United States, but we do intend to generalize this so it could be used on anything, and we're working on that now. Okay. So exercise 2. Hopefully you guys all have Conda and maybe Mamba. And so what I'm going to do right now, which I should have prepared for, but I don't think I did, is to send you guys a file, which is new samples that I want to see, that I want you to try to add to the tree. Use our software. Tell me how many introductions you think there are. And the whole thing is run as a simple snake-make workflow. So literally these commands, git clone, this GitHub, cd usher workflows, snake-make, etc., will produce everything. It should take just a few minutes. And I'm going to take 1 second to send you guys the file you actually need. I think I can send it through here. So give me 1 second. And I just want to see, I mean, this is sort of for me and for you. I want to see how easy this is for you to do with a very little introduction like this, and get some idea if there's maybe a better way for me to do this, or I should think of a more clever way to do this kind of thing. So these are data I downloaded from GizAid. It's a FASTA, so it's a couple megabytes, but it's not very big. Okay, so it should be up. Hopefully you can all see it in the chat. There's a file for you there. We're not going to be able to break into small groups. So basically my suggestion is if you need to, maybe direct message each other in the chat or just try it and see if you can do it. If you can't, that's fine too. But I just want to get an idea how easy this was for you to do and see how you interpret this. And so what will come out of these two workflows is a bunch of files, but the important ones are userseeks.clusters.txt here, which is essentially a summary of the clusters in those samples. And you can actually view the tree at taxonium or taxodium.org. The file that it'll output is a protocol buffer file, userseeks.taxodium.pb. So give it a shot, see if you can answer this question, which is how many clusters do you have in your data? And I'll give you a hint. It's the number of lines in this file, -1. And then where could the largest cluster have come from? So if you examine your tree and you look for the largest cluster, where do you think it might have come from? Okay.
Cool. A small question, is it Windows compatible? Because I do not have my Unix-based laptop.
Yeah, so if you don't have
currently.
that...
Well, I do, but I just have to switch it on. It will take some minutes. Okay.
I've never tried to do this on Windows, so I can't.
Okay, so I think I'm sorry. I think I shouldn't also.
I would recommend you not. Yes, give it a shot. Cool. Again, the sequence file to add is in the chat there. Just like I said, just explore it, think about it, load it up in Taxodium, and look at your tree. I'm just curious to see what you get out of this, and see if you can do it. This will run on a basic laptop. I run it on my laptop all the time. I usually give it 4 threads. It depends how many threads your laptop has, of course. So you'll need to spend some time thinking about that. Cool. I'll be right back. Thanks. I'm back. Did anyone manage to do this? I'm going to call on you, Dimitri, because I got to call on somebody here. So did you manage to do this? Am I talking to myself? Hello? Oh, we've lost Dimitri. Okay, that's not good. Anyone else? No? Okay. I really don't love calling on people I don't know. So I'm just going to go through this. Basically, your questions were, how many samples are in your clusters? To do this, you could just look at that the user seeks.clusters.txt file that comes out. That file lists all the clusters we identified in your samples, assuming that the set of samples you've provided me are the only samples in a given region versus the others. And the reason that's sort of useful is that when I'm a public health officer, I typically just want to know what is the latest and greatest that's happened. Someone's on the chat. Let's see. All right, I've been told I've got to call on Nikita. Nikita, tell me, what did you get done? Nikita? Hello?
I'm sorry, I tried to use this command SnakeMake and I got some error, so I'm trying to install the program.
You don't have Conda?
I have, but unfortunately, I have some mistake.
Okay. While you keep working on that, why don't I spend a minute and tell you why SnakeMake is a great thing? So, and then anyone here can interrupt me the minute they know the answer to this question. So the reason SnakeMake is wonderful is that it can do full workflow management. What that basically means is you can give it a fairly complex command and a series of subcommands, and it will figure out just from that what series of subcommands it needs to run in order to do this. And in fact, it'll actually figure out what's called a directed acyclic graph, a DAG of the jobs, such that it will only start the next job when the previous one has created the correct output. And so it can bifurcate. If you have many cores, it will multifurcate into different jobs as long as they're not dependent on each other. The reason that's really valuable is that when you're running this on a cluster like an HPC environment, or if you run it on Google Compute, I don't know how often you guys use cloud compute, but it can be really, really nice because it plugs into like Kubernetes. And so you can do really, really distributed job management using SnakeMake. And it has this beautiful advantage of it's extremely reproducible, right? The jobs are all sort of hard coded into it. It's just changing the input variables. And so you can never guarantee reproducibility, of course, but you can certainly help enforce reproducibility, which I think is tremendously important, especially if we're going to start making, if we're really going to start making medical and public health decisions based on these kinds of inferences. That is to say, using viral genome sequences or pathogen genome sequences to make intelligible medical inferences, then we clearly need to be able to do to have something extremely reproducible so that we can say, okay, this is exactly why we made this decision. We're sure it works this way. This is the future of medicine, if you will. And so that's kind of a big part of why things like SnakeMake and generally workflow managers are going to be really appealing for these sorts of problems, is that they kind of force it to be reproducible. All right, so did anyone else figure this out in the meantime while I've been sort of stalling for time here? Not looking great. Okay. I'm going to give you guys some homework assignment. Your homework assignment is to get yourselves set up to run Conda, Mamba, and SnakeMake. I'll put those names in the chat so you know what I'm talking about. Conda is the original package manager that was, I think, originally for Python, but we kind of use it for everything now. And then Mamba is a much faster way to install things than Conda. So the very first thing I would do when I install Conda is say, Conda install Mamba. And then once you have that, everything's a lot faster. You also need to install SnakeMake, which Conda can do for you. Oops, sorry, that's not quite spelled right, SnakeMake. And so those three things are going to make your life a lot better if you're doing significant computational development work, because you can build specific environments that are distinct from each other and have different components in them. One really nice thing about the way I've structured the SnakeMake file is that you guys should look into it, how that snake file works. But the reason it's structured well is that it can produce multiple different outputs for one thing, which is quite nice. But it also has this really simple way of flowing through the job, right? I don't have a config file, and it will build the environment for you, right? So that environment file in there, usher.yaml, that's the file that looks like this. Is the reason you don't have to install any of these packages, right? What it does is it says, as long as you have Mamba, SnakeMake will go find all those packages, build the environment for you, and run the whole thing in one shot, which is a really, really incredible thing when you're developing for other people, right? Because literally the only three things you had to have installed would be Conda, Mamba, and SnakeMake, which is really, I think, a huge advantage. I don't know how many of you were developing for bioinformatics 10 years ago, but back then it was always a giant mess of ugly Perl scripts. I was trying to wrangle into some meaningful pipeline and give to other people, and it was just terrible. So anyway, that's a very long way of saying, check this out. If no one got this in the meantime, and it sounds like you may not have, I am just going to go ahead and tell you some answers. And this is a homework assignment now, so try to do this, and I'll ask you next time how it went, and hopefully you'll get the same answer. But the first one is, there are 19 clusters in these data of sizes 11, 5, 3, bunch of 2s, bunch of 1s. This is a pretty normal size distribution. You will always get something kind of like this, which is probably, I guess, geometrically distributed or nearly geometrically distributed. And so it's very normal for introductions to look like this. You get a ton of 1s, a ton of 2s, very few relatively large clusters. If you zoomed in on it, the largest cluster would look, the largest cluster is something like this, where you see a whole bunch of samples from Sweden that we've added. So I've given you data from Sweden. There's a bunch of samples from Sweden. Of course, you cannot definitively infer the introduction, but if you kind of look up the tree and try to figure out where it might have come from, a good guess would be the United States, because all of the closest relatives here are the US. If you look up and down the tree, you'll see more US. And if you wanted to guess within the United States, you might guess Colorado. USA/CO would be the Colorado Department of Public Health. I don't know what the E stands for. So that was kind of the idea behind this. Try to take a look at that again, and I'll be very happy to ask you all about it next time. And we'll see what happens. So at the end here, I'm going to just tell you a little bit about what I think the future of this looks like when we all kind of get going on this stuff and start building out these resources. So the first is that we'll have similarly comprehensive platforms for every major pathogen, right? Major and emerging human pathogen. And this could include even sort of viral isolates that spill over into humans kind of rarely and are very genetically divergent, because it can be extended to deal with other problems. And what I'm showing here is just a quick screen grab of a tree I made for tuberculosis, where you can see that there are a whole bunch of, the same basic approach has worked, right? I inferred this with Usher, actually. And what you can see with the coloring is that I get the exact same lineage structure that other people get. In fact, this tree is extremely similar to others, but the important thing is I'm able to infer and optimize this whole thing in like an hour rather than many, many days. And the other important thing is that it's dramatically different from more typical from SARS-CoV-2, right? So SARS-CoV-2 is a 30,000 base pair genome, and there's millions of samples, so it's a very different problem. This is a bacterial genome, which makes it about 4 megabases in size, which is very big. It's orders of magnitude bigger than SARS-CoV-2. But it also has this cool thing going on where there's a ton more SNPs, as you might expect, right? There's thousands of mutations. And this is just to show you that there's nothing really SARS-CoV-2 specific about any of the work we've done. We've just been focused on it because, you know, it's a global pandemic and it's important to work on it. I think hopefully we'll make real progress. Okay, so what do I think the future of this looks like? The future is going to be doing something like this for every major pathogen, right? So we'll have, in the cloud, we'll live this tree. Every time new samples become available, I will simply add them to my tree using this sort of sequence addition approaches. And it may not be Usher, there may be new things that come out. Probably there will be, that's okay. And we'll store this tree. And then whenever I'm not adding new samples, I'll be sitting around optimizing my tree. And I cut those slides from this, I'm pretty sure, but just to tell you what optimization is basically that every now and again, you get it wrong, right? You add a sample in a place that is suboptimal in the global topology. Of course, I'm sure you all know this, but tree space is disturbingly complex, right? It's incredibly complex. There are tons of different possible arrangements of the lineages. I don't even remember the math for it, but it you know, even with 10 samples, you can make a lot of different trees out of that. So you could never search everything. That's, you know, it's NP hard to find the perfect tree, and no one can do that. So what you instead do is simple little optimization heuristics like this one, where I would prune out sample F and try finding a better location for it. In other words, a place that would minimize the number of additional mutations that are required. In this case, it might be here where it joins up with sample E, and we can see that they kind of go together down here. And then you would repeat, actually. So you essentially, anytime I'm not adding new sequences, I'm optimizing the tree. My algorithm may run to completion because I'll run out of tree moves to try. That's fine. And then it'll just repeat. It'll go back to the beginning, and when new samples come in, we'll start adding and optimizing again and again and again. The idea is that by hosting these for every major and emerging human pathogen, public health offices and doctors will be able to check out the latest release of the tree, and they'll be able to add their samples in seconds. And they can rapidly optimize these local subtrees. So once they add all their samples, you'll be able to optimize your local subtrees sort of on the fly as well. And what you should get out of this is sort of a nearly immediate and highly reproducible output, right? So in other words, I would like to be able to get out of this something that is extremely fast so the doctor or public health officer can make intelligent decisions, but also you would like to get out phylogenetic annotations. And what those are ways of telling public health officers and doctors what is important about this placement, right? So if I added sample F, and I already knew that samples D and E had some important antibiotic resistance or something else that would affect the treatment or public health measures, I want that information to get back to them. So what they'll do is they'll take this information from these and propagate it out to sample F. And I have exactly that here. So essentially the idea behind this we're working on right now is a CDC proposal called that I'm tentatively titling "Everything Needs to Be Faster." And so the idea is there's these strain-specific annotations that we'll have. So some are already known, right? For example, in tuberculosis, there are many, many, many strains that are known to have various different amounts of antibiotic resistance, right? And it's annotated all over the tree. If you go out to, but of course, we also need to be able to generate those annotations. So the simple thing is I already know, right? I might know sample D is resistant to some important antibiotic. And then when I add sample E to my tree, I propagate this information from D to E, and I say, okay, we know that you are resistant to this antibiotic, and so you need to have this we need to tell the doctors that we should look out for that. On the other side of this is something like this, where what we really also need to be able to do is take sample E and annotate it. Right? So there may be many strains of E, or many, many different individuals all closely related to E, that are treated in different hospitals, and they'll get different treatments. And with a linkage to medical records, which we really need to have, with a linkage to medical records, you would be able to actually generate these evolutionary annotations as well, right? The idea being that, okay, maybe I treat 1,000 people that all have close relatives of E, then one treatment just always does much better. I would like to put that information back into my tree so that the next doctor that sees a sample that looks a lot like E can say, this treatment is the one that worked previously for a strain like this. There's really good idea that this is the way to go. This is where we need to be going in order to treat these individuals. So this is a huge, endeavor. It's going to take years to reinvent public health and medicine, but I'm really excited about it. I think it's also tremendously important to start thinking about these things and get this information in here. So some reminders for next time. So next time I'm going to tell you about, so as I'm wrapping up here, before I take any questions, I'll just tell you about some reminders for next time. Then I'd like to take some questions. I'm hoping you guys have some, and we can go from there. But some reminders for next time are that, remember, we're going to talk about selection and we're going to talk about recombination. So there's a few things. One is, remember this observation from the first class, this sort of genomic and subgenomic RNA production observation. So the genome is this whole thing on the top here. And it's replicated by making the negative sense copy of this positive strand and then making positive copies off that negative strand. So it's just replicating itself, going back and forth along the genome. Down here are the subgenomic RNAs it produces. And what these are is sequences. So there's this translation regulatory sequence here. It gets spliced onto these things in a few different places. And what will happen is you'll get you get these proteins translated this way. So this would make this light teal protein here. This makes E, this makes M, this makes the spike, a few other things. And so what that means, among other things, so this is the splicing pattern that we showed you last time. This is how you can measure it. What that means, among other things, is that you'll have more and more RNA toward the back third of the genome, right? You just have, because you need to make proteins like this, but the transcript for ORF1A also includes all the spike and all these others, right? It goes through the whole thing. The spike is this chunk here, and so it starts at spike and goes through the rest. The same is true here, here, and here. So the idea is you're just always going to have a higher cumulative amount of these subgenomic RNAs toward the back third of the genome, because every preceding transcript contains all these final stuff as well. Okay. The other thing that I want to remind you about, there is an absolute ton of genetic diversity in and around the spike protein region, right? So your spike is here, this chunk, and it pretty obviously coincides with this enormous dip in sequence similarity, which is an opposite way of saying an enormous peak of sequence diversity among these viral genomes, right on top of the receptor binding domain here, which is sort of this line more or less. And so we strongly suspect this is a major site of evolutionary innervation in the virus. And it's something we really need to be studying both in the context of natural selection, but also in the context of detecting and understanding recombination, because if recombination is making more variation by mixing haplotypes in these regions, that's actually tremendously important and something we need to be thinking about, monitoring and understanding sort of as quickly as it happens in real time. Okay. All right. With that, I would love to take any questions you guys have about this stuff. And again, you have homework for next time, so tell me the answer to my question. No one has any questions? Okay. All right.
Oh, maybe that's because it is quite late here.
Yeah, no, I get it. I'm sorry, guys, for making you stay up with me. It's okay. Next week, I will be at the normal time, I promise. So I'm ready. Cool. Okay. Well, no worries. Go to sleep.
I have a question. I'm sorry.
Oh, good.
Yeah, it's regarding the exercise, the last one. I'm just curious about SnakeMake, because unfortunately I couldn't install it using Conda. For some reason,
You can't install.
it can't search the package.
There's a way to do this. Sorry, you may need one additional instruction from me to do this. There is a way, I assure you. Let me find it real quick. You need to, oh my gosh, sorry about this. You need to add a channel for Conda. So do you already have Mamba installed? Well,
yeah, I do.
That command ought to do it. It may also work if you do Mamba install instead of Conda install. Let me know if that doesn't work for you, though. Basically, what I forgot to mention is, that there's certain channels, right? In other words, there's the basic Anaconda channel, but then there's also Bioconda, which is where a lot of these things tend to end up. So SnakeMake, for example, is one of them, or maybe in Conda Forge, I forget exactly how it works.
Yeah, I've tried to use channels. I know about them, but unfortunately, well, I added a Bioconda, and it didn't work. Maybe if I add Conda Forge too, it will work. I'm not sure.
Shoot. Okay, I definitely did this without Conda Forge, but try it and just see if it works for you real quick, and we can go from there. What kind of computer are you using right now? Are you on a PC or a?
I'm using laptop.
Is it your operating system? Is it Linux or is it Windows?
I have Windows, but I have inbuilt core for Linux.
Subsystem. Yeah, the Ubuntu subsystem. That's actually really nice. It made Windows usable for bioinformatics, where it used to be worthless. So that's good. I'm trying to figure out, that's definitely a command I've run before, so I'm a little surprised this isn't working for you, but there may be some other way you got to do it. I don't know.
Yeah, I think we can download it by using sudo apt-get.
That should also, yeah, if it's in the aptitude repository, that'll work as well. I typically like to use Conda for these things because it's, not everyone's on Ubuntu, so it's a little harder to give instructions that way, but it should work. Yeah, okay. Who else has questions? Anybody? Did anyone else get this installed, or do I should have given you way more advanced notice on this? My apologies.
There is a question from Dmitry. He watched us on YouTube, actually. Had some problems with his own. So his question is, how are systematic errors detected? Are there any deterministic algorithms?
No. So the way we did this in the end, we spent a lot of time staring at this and trying to make harder choices. The one algorithm that you can use is what we called that line I showed you, the extremal. All right, so I definitely just installed SnakeMake that way. I don't know what to tell you, but so if I go back to that, I'll show you what we did. There is an algorithm, and you can get this from Yatisha's GitHub package on this stuff. This one. But basically, if I go back to that slide and I try to show you this, for the record, I think systematic errors are probably very important, and they're going to be more important as data generation becomes increasingly widespread and disaggregated. But the idea was, if we had, if you could detect this line, this extremal line, you could certainly get rid of the worst of the systematic errors. And so what I think this line represents is essentially how frequently you would group two samples or more based on a single mutation thrown down completely at random on the tree. They're of course not completely random, but let's imagine a site was truly non-heritable. It would pretty much be somewhere on this line. So that we called that the extremal. And we wrote some pretty simple algorithms to basically go along and say, okay, let's pick out anything that's the furthest to the right or the furthest up and above or near to this log2 line that we fit to the edges here. So you can check that out in our paper, which I'll find you right now. Let's see. I'll just go to my Google Scholar. That's easy. It's this one, where we really dug into this, I think, fairly deeply. There's been a number of other papers along these lines. Ours is really about this, about the specific case where, the chat would be above me, of course. Sorry. Dimitri, check that out. But basically, that's where we did this. It's a simple algorithm. I would like to do more complex things. I think there may be better ways to do this stuff. And I think it's going to be really important that we do figure out how to do these things in simple, generalized ways that we can reproducibly use across a pretty broad range of taxa and species. The big issue, of course, will also be the specific molecular assays that we develop, because those assays may also introduce systematic errors that aren't necessarily associated with the lab, they're associated with the assay. And so this is, the better metadata we have, the more we'll be able to do with this kind of thing. And I think it actually is, it's one of these things that may not sound super interesting, but I think is incredibly important to think about, is how to handle these kinds of problems. Great question. Dimitri, did that answer your question, or do you have something else you'd like to talk about? Or sorry, he's not here. He's asking somewhere else. My apologies.
Yeah, he'll reply me.
Okay.
Maybe with some 20-second delay.
Yes, yes. Makes sense. That's all right. Okay. Okay. Cool.
No, I don't get anything from him.
Okay. We'll have to come back to it some other time, guys. So again, for next time, do that exercise. See if you can figure it out. I think you probably can. I would really love feedback on how straightforward this is to do for somebody who's never had to do it before, right? Because the thing what we're trying to do is to put this into the hands of public health officers and medical practitioners and get them to be able to do this sort of thing. Obviously, we're not there yet, but I feel like you guys are a good intermediate level of who has some skill in this stuff but hasn't done this exact thing and can tell me how hard it is or not. So clearly it was harder than I thought because the installation is hard. So my apologies. Get your Conda, Mamba, and SnakeMake installed. Let's go from there. Cool. Thanks, guys.
Thank you very much, Ross.
All right, I guess I'll see you all next week then.
Yeah, see you in a week.
You guys, bye.